We are talking about Lac Operon by Superna Bajaj, Lauren Enriquez, Crystal Leo, and Josh Sosa.
Okay, so I'll begin by explaining the Lac Operon components. The repressor gene is LAC1 gene. This gene goes through transcription and translation to create the LAC repressor. For the regulatory region, which is right here in this area, we have the promoter and the operator labeled as P and O. The promoter is the region of DNA that initiates transcription, and the operator is the segment of DNA which transcribes factor proteins. The lactose utilizing genes, which are right here, and the structural part of the genes. So LAC-Z is what helps create beta-galactoside. LAC-Y is what helps create permase. And LAC-A is what creates transacetylase. Next, we will be discussing regulatory proteins. To begin, we'll start with the LAC repressor. The LAC repressor binds to the operator, preventing transcription from occurring. It is created by the LAC1 gene. It is always made, will express, and it responds to lactose, which inactivates it. Next, I'll be discussing the CAP-CAMP complex. CAP stands for the catabolite activator protein, and CAMP is cyclic AMP. CAP is controlled by cyclic AMP, meaning CAP must be with cyclic AMP to turn on. Cyclic AMP production is inhibited by glucose, so in high levels of glucose, you're going to have low levels of transcription. And low levels of glucose, you'll have high levels of cyclic AMP, which will have high levels of transcription.
Now we're going to talk about controls of Lac Operon. So when glucose is present and lactose is absent, there's going to be no transcription because there's no CAP-CAMP complex and the repressor binds to the operator. When glucose is absent and lactose is present, the transcription will be on because the repressor cannot bind to the operator and the CAP-CAMP complex is available. And when both are present, here, the transcription will be on, but it will be weak because the CAP-CAMP complex does not form and the repressor cannot bind to the operator.